Everybody could attest this virus was a futile quest. COVID made us all depressed. We were so lonely. Our social life became lonely. Right now we're hopeless. We are almost helpless. Every time we went outside, someone was sick. We were stagnant and idle. We were stuck in our wormhole. Then we had mRNA to protein sub you into vector-bats. Then this virus has crashed. When you get back, yeah, there's no need for cash cause it comes from your tax. The vax, the vax, the vax. So you have no idea how any of these things work. Don't worry, I got you. So mRNA, protein symbiote, and vector vaccines are all three different kinds of vaccines that we have used to make what we see as COVID-19 vaccines. So let's start off with vector vaccines. It uses the central dogma, which is DNA to RNA to protein, to go through the whole traditional process of making a vaccine. So what we do in the Johnson & Johnson, which is the vector vaccine, is that we take the DNA from the virus, or the genetic information, we inject it into your body, into your cells, which are then processed so that your body is able to make the proteins and display them on the cell. When they are displayed on the cell, then immune system cells can come, remember that protein, create antibodies, so when the real thing comes, it can protect against it. Now, Johnson & Johnson is only 66.3% effective. However, we'll get into more effective ones later. Next is mRNA. So mRNA, kind of similar, except it skips the whole DNA part, and we just directly inject mRNA into your blood, into your body. So how this works is, since the mRNA comes, it can just be directly put into the ribosome without any transcription, and it can be a protein. And so once it's a protein, it can go through on the outside of the cell membrane, immune system, whole shebang. And we see Pfizer and Moderna, both mRNA vaccines. Pfizer is 95% effective and Moderna is 94.1% effective. We're liking those numbers a lot more. Now, the last one is protein subunit. So this is, again, same process, except it skips the first two parts. Instead of using both DNA and RNA, it just skips straight to the protein and it injects the proteins in your body. Then the cells are able to process that protein and consume it, so it can show them on the outside. And the same process happens where your immune system detects the proteins, and they can create antibodies and fight against it. That is an example of Novavax, which is the newest vaccine, and which is 90% effective.
Vaccines go through a thorough research and testing process before they can ever be released to the public. According to the CDC, vaccine production follows these general steps: exploratory, preclinical, clinical development, review and approval, manufacturing, and quality control. Additionally, in the US, all vaccines must be approved by the Food and Drug Administration before they are released to the public. The general process for approval begins with the exploratory stage, where research takes place. An idea is formed of how the pathogen infects the body and causes disease. Before it is tested on people, the vaccine goes through the preclinical stage, where further research is performed and animal testing can take place. If the product is unlikely to cause harm in people, it can proceed to the clinical development stage with the FDA's approval of an investigational new drug application, where all research is compiled and it is decided if the test took place according to good laboratory practices. There are 3 phases to the clinical trial. Phase 1 is with a small group of randomly selected volunteers who are all healthy and have never been infected with COVID. This phase is to test for any adverse reactions or unwanted side effects. Phase 2 includes a larger group of volunteers from different demographics who may have underlying health conditions. Information on effects of dosage and immune response is collected at this stage. This stage will also include a group of people who the vaccine is meant for. Phase 3 is with a much larger group of volunteers to test the effectiveness and give more information on immune response. Once there is an effective and reliable manufacturing process, a biologics license application is submitted to the FDA. It is essentially a document that compiles all the research and testing information on the vaccine to show that it is reasonably effective and safe. The FDA makes its decision based on the risks and benefits with a collaborative team of experts in the field. Sometimes preliminary approval for use is granted in emergencies before the testing phase is complete. Emergency use authorization may be granted by the FDA in special cases like the COVID pandemic. Requests can be submitted during phase 3 of the clinical trials when extensive research and human trials have already taken place.
We have discussed what goes into the process of making and testing vaccines. Now, let's talk about what happens when this process is undergone and the results do not make it to the next stage. What is an example of a COVID-19 vaccine that was initiated yet didn't succeed? Let's get into it. There is one COVID-19 vaccine that was found to show less protection in the region of South Africa compared to other countries. This is due to a SARS-CoV-19 variant that is able to dodge key antibodies and has become widespread. According to an Oxford University trial for a COVID-19 vaccine produced by AstraZeneca, which ran from June to November of 2020, it was found that starting 2 weeks after the second dose, when the participants presumably were fully immunized, 19 of mild to moderate disease developed among the vaccinated, versus 23 in the placebo group, resulting in an efficacy of 21.9%. Ultimately, this was far below the 50% minimum required for emergency use authorization in many countries like South Africa. AstraZeneca began South African small efficacy vaccine trials conducted with about 2,000 people. The results found such low efficacy against mild and moderate disease, under 25%, that it would not meet minimal international standards for emergency use. These COVID-19 findings are reported by Science.org, which is supported by the Heising-Simmons Foundation. Similarly, health authorities in Denmark, Norway, and Iceland have suspended use of AstraZeneca's COVID-19 vaccine following reports of adverse effects. It is currently not possible to conclude whether there is a link and that it needs to be thoroughly investigated. However, it is a cautionary decision. Those are some of the facts about what goes into making sure that a vaccine is suitable for public use. The priority of its design is to prevent serious complications and death. This is not a game. You can end up dead. Now my friend will go more into detail about the impact of variants.
Well, it's not the strength of a vaccine or how much of an immunological response it causes. It is how much less of a risk someone has of developing the disease if they have taken the vaccine. We care mostly about variants of concern. These are variants that the WHO has designated as having significant detrimental changes to transmissibility, clinical disease presentation, or effectiveness of public health or social measures. As of March 7th, 2022, these include Alpha, Beta, Gamma, Delta, and Omicron. Each of these variants was first tracked on a different continent. Alpha was documented first in the UK, Beta in South Africa, Gamma in Brazil, and Delta in India. Omicron was first documented in multiple countries. In addition to variants of concern, there are also variants of interest. Variants of interest are those with mutations that could potentially affect important characteristics of the virus and are also identified to cause significant community transmission or to transmit in multiple clusters. These include both the Lambda and Mu variants, which were first documented in South America. The effectiveness of COVID-19 vaccines against asymptomatic infection has been shown to be reduced when dealing with newer variants. For example, although COVID-19 vaccines are highly effective against asymptomatic infection for the ancestral virus and Alpha variant in the short term, they are reduced by around 20% against Delta. Well, it's still important to get vaccinated because vaccination significantly reduces your risk of developing severe illness. Vaccination is still the most effective defense against this disease by far. Plus, these new variants of concern make strategies that are purely aimed at stopping transmission, such as mask wearing and social distancing, unlikely to have more than a short-term effect on preventing severe COVID-19. It's unlikely. You see, unlike other viruses such as measles, coronaviruses are likely to continue to circulate seasonally because infection with them does not lead to durable immunity. This would mean that we might reach a point in which, just like the flu vaccine, we must get our yearly dose of a coronavirus vaccine. Well, some recommended courses of action when it comes to dealing with this shift include prioritizing prevention of severe disease, In areas of low vaccine availability, we should prioritize delivery of the first dose of a vaccine, and in areas of high vaccine availability, we should prioritize delivery of 2 doses in the elderly and immunocompromised. Overall, we should really focus on creating more global equity when it comes to vaccination as a means to stop the spread and ensure that severe disease is always the primary thing to be prevented everywhere.